Every research project ends up scattered, papers in one folder, notes in another, results somewhere on a shared drive, and a thesis that has to pull it all back together 18 months later. ResearchVault keeps the whole thing in one place, on your own machine. There is no account to make and nothing is uploaded. Your vault is a single folder you can see, copy, and back up like anything else that matters. Here is what that looks like on a real project. We are going to build a biomarker study from nothing. A new project starts by choosing a template, and the template is the important part. It is not a theme. It decides the sections this project will have, and the questions every experiment will ask. Biotechnology gives us research question, literature review, hypothesis, protocol, sample tracking, results, analysis, and publication. A social science template would give a different set, and so would one you write yourself. And the template is copied into the project, not linked to it. Editing the template next year will never reshape work you have already done. The tabs are where the thinking goes. Research question first. Which serum protein panels predict early recurrence, and why does it matter that we find out? Then the hypothesis, a panel of 8 proteins, locked before we look at the validation samples. That word, locked, is the whole design. Then the protocol, 2 cohorts, recruited at different sites, assayed the same way. Each section carries its own progress, and the overview adds them up. Nothing here is a form you have to finish. It is a document that fills in as the work happens. The experiment log is where the template earns its keep. Every run here answers the same 6 questions: objective, method, materials, observation, result, conclusion. Not because a form insisted, but because the biotechnology template said that is what a wet lab run records. Run 2 is the interesting one. Signal falls across the plate from column 1 to column 12, regardless of which patient is in the well. That is not biology. That is the plate. Run 3 fixes it without touching a pipette. Same samples, plate position modeled as a covariate, and an 8 protein panel comes out at an area under the curve of 0.84. The failed run stays in the log. It is the reason the next one worked. Every claim in that analysis came from somewhere. References live in the vault rather than in a separate reference manager. Articles, books, reports, and links are all the same kind of thing here, because a citation does not care what shape its source was. The second entry is the one that explains run 2, plate position artifacts and high-plex proteomic assays, Nakamura, Nature Methods, 2023. Read before the batch effect appeared, which is why it was recognized instead of argued with. All of it exports as bibtx or RIS. The bibliography is a command, not an evening. Not all research happens at a bench. Fieldwork record sites and the visits to them. This one is a biobank at a regional cancer center, with its coordinates, and a note that the freezer log is on the wall by the door. The person who visits next might not be you. Each visit carries the conditions: who was seen, how many people consented, and how many declined. That last number is the one people forget to write down, and the first one a reviewer asks for. Research runs on dates, and the dates are never all in one calendar. Tasks carry subtasks. Unblind the validation cohort is 3 steps, and the first of them, confirm the panel lock is documented, is the one protecting the result. The calendar merges what would otherwise live in 3 places: meetings, deadlines, milestones, and every task with a due date in 1 month. The timeline puts the whole program on a single bar, so a supervisor can see where 18 months actually went. Eventually, the work has to leave the building. Submissions follow a paper through its life. This one went from research to draft to submitted at Nature Biotechnology, and the board keeps every date it moved on rather than only where it sits today. The thesis gets its own workspace, because a thesis is not one document, 6 chapters, each with a word target and a status of its own. Introduction done. Literature review and methodology and revision. The validation cohort still an outline, because the samples are still on the plate. 31,000 words written against a target of 58,000, and the number is counted rather than guessed. The last problem is finding any of it again. Topics are tags that work across everything. Biomarkers sits on the project, on the methods paper, and on every experiment, so one click gathers work that lives in 5 different screens. Search reaches inside the content, not just the titles. Ask it for plate position and it finds the project section that recorded it, both runs where it showed up, and the paper that named it, with the phrase picked out in every result. Full text, across the whole vault, instantly, which is what you get when the whole vault is one file on your own disk. One last thing, and it is the reason all of the rest works. Your vault is a folder in a place you chose. There is no account, no sync service, and nothing leaves this machine unless you move it yourself. A backup is a snapshot written into a folder you pick. If that folder happens to be your Google Drive or your OneDrive, then you have cloud backup on your own terms, with nobody in between. ResearchVault is free, with one small advert on the dashboard. Removing it is a single purchase, once. Nothing else is locked, and nothing expires. Your research outlives the software you kept it in. That is the whole idea.